இது நம்ம ஐயா கோச்சிங் சென்டர். வாங்க படிக்கலாம்.
இந்த வீடியோ உங்களுக்கு பிடிச்சிருந்தா லைக் பண்ணுங்க, ஷேர் பண்ணுங்க, சப்ஸ்கிரைப் பண்ணுங்க. Thank you.
We'll see application of linkage. The distance can be identified. Distance can be identified by the incomplete linkage. Distance can be identified by the incomplete linkage. And the unit is centimorgan. Distance between gene is measured and the unit is centimorgan. You can see here the strength of linkage. Strength of linkage is inversely proportional to, inversely proportional to distance between linked genes. Distance between linked genes. And it is directly proportional to crossing over. Directly proportional to crossing over. Next we'll see genetic map. Genetic map or linkage map or chromosomal map. See, in genetic map, different genes are linearly arranged. Different genes. How these genes are arranged? Linearly. How these genes are arranged? They are arranged linearly according to percent of recombination between them. Percent of recombination between them. Then with the help of what is the use of this genetic map or linkage map or chromosomal map? With the help of this map, we can find out the position of a particular gene. We can find out the position of particular gene on chromosome. Particular gene on chromosome. Then the genetic map is helpful in the study of genome. It is helpful in the study of genome. Complete linkage. You can see one example. See the parent, red eye, male Drosophila with vestigial wings, purple eye, female Drosophila with the long wings. See, this is capital R, small l, capital R, small l. And this is small r, capital L, small r, capital L. That is red eye with vestigial wings. This is purple eye with long wings. When these two homozygous parents are crossed, and this is the gamete, capital R, small l, capital L. And the F1 generation, the F1 progeny is red eye with long wings. Red eye with long wings. That is capital R, small r, capital L. Next one. That is previously we have seen when we crossed this red eye vestigial wings male Drosophila with the purple eye long wings female Drosophila. The F1 progeny is red eye long wings. Now we are going to see test cross. In test cross, the red eye long wing is crossed with purple eye vestigial wings. See, this is heterozygous. This is what? Heterozygous. Small l, capital L. And this is pure purple eye. That is purple eye. That is recessive. The parent of this recessive. Now we can see this test cross. When these two parents are crossed, see the gamete formation, capital R, small r, capital L. And here only one type of gamete, small r, small l. When this gamete is crossed with this one, then we will get, there is capital R, small l, capital L, and when this is crossed with this one, small r, capital L, and small r, small l. Now we can see the offspring exhibit only which character? Parental combination. The offspring exhibit only what character? This is red eye. This is red eye, vestigial wings. Red eye, vestigial wings. And this one is purple eye, long wings. So, purple eye, long wings. Long wings. So, these exhibit only what character? Parental combination. No new combinations are formed. These are due to complete linkage. There is no crossing over. Thank you.